Now here students, I'm going to continue with this one topic, that is the unit of life mind map. Okay, so see what here I'm saying that whenever I am discuss about the cell, it means that what the cell basically, there are various types of the cell, but on the basis of the nucleus there are two type of the cell, my dear. Number one, that is what prokaryotic, and second one is what eukaryotic. Now, the point is here to know here that what is prokaryotic and what is eukaryotic, right? So see, before we have to be know about the various cell organelles, whatever the present in a cell, first of all, we should have must to clear here the point that about the prokaryotic and eukaryotic, my dear. Okay, so see, just here I'm going to tell you that about the prokaryotic and eukaryotic also. You know that the pro means what primitive and karyon means what nucleus. So it means that what the organisms which had what primitive nucleus. It means that the organisms which has not well-defined membrane-bonded organelles, right? So if the organisms which do not having a membrane-bonded organelles, then it is known as what prokaryotic, right? Take an example of that very prokaryotic. The example is what bacteria, virus, PPLO, that is also a smallest microorganisms, pleuropneumonia like organisms, that is mycoplasma, they all are what prokaryotic, right? Now, whenever I'm going to tell you about the eukaryotic, the first thing that is eu means what true and karyon means what nucleus. So it means that those organisms which has true nucleus, it means that the organisms which has well membrane bonded organelles, my dear. So we all are multicellular organisms, complex organisms. So that's why I'm saying here we all are what eukaryotic. And you all know that whatever the plants and animals, they all are what eukaryotic, my dear. Okay. Now see, this one is the thing that we have to be go through only a short details, because this one is the mind map. Okay, so here I'm not going to discuss in the details. So what the things is be here given in mind map, that only things you have to be know about. Okay, let's see. After that knowing about the types of cell on the basis of the nucleus, then after here the point is given what cell theory. See, this one is the point given here, that is what cell theory. Clear? So here on the basis of the nucleus, the two types of cell, prokaryotic, eukaryotic, is clear. Now here the word cell theory. Now the point here to know about what cell theory. Let's see. If I'm saying here about the cell theory, this one is the second point, cell theory, then what we know. Let's check here. Wait a moment. Yeah, here I'm going to write what? Cell theory, my dear. Check. Cell theory. See, if I'm going to tell here about the cell theory, it means what? Cell theory, that the theory which is given by the two scientists basically, and one of the other scientists also. Basically, you say the three scientists. Number one, that is Matthew Skaldian and Theodor Sion. What I'm going to write here, I'm going to write what? Matthew Skaldian. One is botanist, another one is what? Geologist. So Matthew Skaldian and Theodor Sion. And what I'm saying, Theodor Sion, my dear, they gave what? They gave cell theory. But here, only these two scientists gave about what? The life which is present in a plants and animals. Clear? So here only the Matthew Skaldian and Theodor Sion suggest us that what? All living organisms are composed of cells. Here the two scientists what only say about this one thing only, all life, either the plant and animals, all living organisms, you say here, all living organisms composed of what? Composed of cells, my dear. Clear? See, this one is the thing, all living organisms are composed of cells. Either the living organisms is plant or the animals, that all things is made up of what? Cells, my dear. And this thing is given by the scientist, that is what Matthew Skalden and Theodor Sion. But now the question is here. Now the question is here. What about the question? That is, What about the question is here? Here in this theory, not give in details about that. Um, why I'm saying this? The all living organisms is composed of cell. This one is the thing. Getting? See, one of the scientists, one of the scientists that is named, that is named, that is the name Rudolf Virchow. Hope so you know the name. Right here, I'm going to write here Rudolf Virchow. See, Rudolf Virchow gave here that gave here that what you know, that is that is all cell arises from what I'm saying here, all cell arises from what, all cell arises from pre-existing cell, pre-existing cell, my dear. See what I'm saying to you. See what I'm saying to you. Listen carefully. Whenever anybody can ask you about the cell theory, then what you say? About the cell theory, you can say that the Matthew Skaldian and Theodore Sion, my dear. What I'm saying? Matthew Skaldian and Theodore Sion, all living organisms is composed of what cell, my dear, right? Getting what I'm saying? So all living organisms, either the living organisms is what, plants or the animals, they all are what, made up of or composed of what, my cells. Clear? Now the question is here, um, whatever the Matthew Scully and Theodore Sion gave about the organisms, that is living organisms is made up of cells, you know, they doesn't clear here that what the cell is composed of, how the cell is composed of. So after that, the scientist Rudolf Virchow suggests that all cells arises from pre-existing cell. Here I'm going to give you one example to show you that how I'm saying here that the all cell arises from pre-existing cell. Correct? Let's see. Suppose if I'm here giving a one example here, let's check here. Suppose I'm giving you example here. Suppose how we say that all cell arises from pre-existing cell. Suppose here I'm giving you example. One is that is what, male sex cell, that is a sperm, unite together or fuse together, female sex cell, that is ovum, then it gives what, my dear, zygote. Clear? So sperm is what, haploid cell? Definitely, sir. Ovum is also haploid. Clear? And this zygote is what, diploid. Am I right? Yes. So see, if I'm saying here about that the sperm unite with together what, ovum, then it form what, zygote. So see, sperm is a cell or not? Yes. Ovum is cell or not? Yes. So after this, sperm and ovum combine together and it forms zygote. Yes or no? Yes. So this one is also a cell, but this cell arises from which one? Pre-existing cell, na? Getting my point or not? So here I'm saying that the pre-existing cell, it means what? Whatever the cell we see right now, that all very cells are existing from previous cells. Getting my point or not? So that's why here, if anybody can ask you that how many cell theory, then what you say? There are two cell theory, my dear. Two cell theory, my dear. Getting what I'm saying? Getting two cell theory. Getting? So one cell theory is what? All living organisms composed of cell. This one is the first cell theory, and second one is what? All cell arises from pre-existing cell. But the difference is here that the two cell theory that is given by the three scientists, name one first Matthew Scaladian, second one Theodore Sion, one is the botanist, second one is what, zoologist, and third one is what, Rudolf Virchow, my dear. Okay, clear? So hope so this thing is clear. So about the cell theory, check here. About this one, cell theory is clear. Yes. Now, next point here to know about the word endomembrane system, my dear. You know what the meaning of endomembrane system, my dear? You don't know. Let's see. if I'm going to tell you about the endomembrane system, it means what? It means what? That very system which is contained within the cell membrane, which having a what? Membrane bounded organelles, my dear. You are not getting. See what I'm saying here. I'm going to tell you about with the help of diagrams. See here. This one is what? This one is what? Nucleus. Suppose this one is what? Nucleus. And this one is what? Cell. See what I'm going to tell you. Clear? So this one is which type of cell? This one is what? Animal cell. This one is what? Animal cell. Right? So if I'm saying here that this one is animal cell, see what I'm saying. I'm saying that here, wait for a moment. Inside the cell membrane, there is what? Dense fluid. The name of the fluid is what? Cytoplasm. Okay, this one is what? Cytoplasm. Right? Now the question is here that how we say that endomembrane system, my dear. Endomembrane system, it means that the organelle which has what? Membrane bounded organelles. So see, if I'm saying here that endomembrane system, the system which are interrelated with each other also. See what I'm saying here, interrelated. How the organelles, one of the organelles is what? Interrelated with each other. See here. See, this one is the nucleus and this one is ER. You know the meaning of ER? That is endoplasmic reticulum. See what I'm saying here. This one is what? ER, endoplasmic reticulum. So you viewing this thing or not? Yes, this one is what? ER, endoplasmic reticulum. And after that endoplasmic reticulum, they form what? Yes, that is GB, Golgi bodies. What I'm saying? Golgi bodies. And after that Golgi bodies, they form what? Vesicles. See, this one is what? Vesicles. So see what I'm writing here. Here I'm writing here is what? This one is what? ER, endoplasmic reticulum. And this network of endoplasmic reticulums, after that, this one is what? GB, that is what? Golgi bodies. One phase is cis phase and other phase is what? Trans phase. And this one is what? This one is what? This one is, you say, this one is say you what? Lysosomes. What you say? Lysosome. Specialized cells, suicidal bag of cell, you say? Lysosomes. When we say the lysosomes, when it move outside the cell membrane, but here it is inside, na? So you say here what? Say here what? Vacuoles. What do you say? Vacuoles. Whenever inside the cell membrane, but when it goes outside the cell membrane, then it is known as what? Yes, then it is known as what? Suicidal bag of the cell. You know, that is what? Lysosome. What I'm saying? Lysosome. So when I'm saying lysosome, whenever this one move out from the cell membrane. Getting my point? So see, if anybody can ask you about the endomembrane system, it means that a system which are interconnected with one organelles over another organelles. You're not getting my point. See, if I'm saying here that this one is what? Nucleus. See, check what I'm saying here. Listen carefully, then you understand that what I'm saying. Okay. So see, this one is the nucleus, and you all know that the nucleus is the what? Brain of cell, right? So whatever the things occurred, that is suppose synthesization of protein. Okay, so synthesization of protein where take place, you know, that ribosomes. Where the ribosomes present? Ribosomes present on the surface of ER, right? So if the ribosomes present on the surface of ER, then it is known as what? RER. Am I right? That is rough endoplasmic reticulum. But if the ribosomes is not present on the surface of ER, my dear, then what we say? SER, smooth endoplasmic reticulum. Yes or no? But here I am written only what? ER, ER of two types, RER and SER. So RER when I am saying RER then whenever the ribosomes attach on their surface, getting, but whenever the ribosomes is not attached on the surface, then it is known as what? What? SER. So here the ER And on the surface of ER, there is a what? A ribosomes, and that very ribosomes, see, suppose on the surface here is there is what? Ribosomes. This one is what? Ribosomes. Clear? So ribosomes what move where? Inside the tubular structure that is known as what? ER, in a ER tubule. Then after it passes through where? Golgi bodies, through cis phase. Then after it passes where? Move outside from Golgi bodies, known as what? Vacuoles. Right? So this one is the thing. So after this one, the transferring of one organelle to another organelle, and they are interconnected with each others. Getting my point or not? So see, if I'm saying here that the endomembrane system having what? Continuous membranous system, which is organized in a sequence manner that coordinate with one another. Getting? Coordinate with what? One another. That is what? ER coordinate with GB coordinate with what? Vacuoles. Right? Now, the question is here, endomembrane system is such a system which are interrelated with each other. Very good. So what the things is interrelated here and what the ordinals is here? You see that in a endomembrane system, here is what? Lysosomes. The first time saying about what? This one is what? Lysosomes. See, this one is the lysosomes. So lysosomes, my dear, known as what? Suicidal bag of cell. What I'm saying, lysosome is what? Suicidal bag of the cell. See here, what I'm going to write. See here, I'm writing here what? Lysosome. If I'm saying here about the lysosome, that is what? It is a suicidal bag. Why I'm saying it is a suicidal bag? Why I'm saying here that it is a suicidal bag? Yes, lysosome is suicidal bag because it it engulf all the all the all the harmful substances whenever invade inside the cell. Invade inside the cell. See what I'm saying? Invade inside the cell, my dear. See what I'm saying? Suppose this one is the cell. This one is the cell. This one is the cell. And inside the cell here, that is one. That is what? You say this one is what? Nucleus. This one is what? Nucleus, having double layered membrane. I will give you in details in detailed classes. And this one is what? Cytoplasm. Clear? Say this one is what? Cytoplasm. Suppose if any harmful materials goes invade from outside to inside. See, from outside to what? Inside. So suppose if I'm saying here that the harmful materials such as bacterial, fungal, or any harmful materials invade inside the cell membrane, then you know what? Here the lysosomes, here the what? Lysosome, that harmful materials engulfed by this very lysosomes and self-destroy, but lysosomes protect other cell organelles. Protect what? Other cell organelles. So self-destroy or self be what did, but that doesn't make them harm of another organelles, my dear. So that's why I'm saying here that the lysosomes is what? Suicidal bag of cell. So self become dead, but they protect from what? They protect which one? Other organelles. So this one is very short way I'm going to tell you, but whenever I'm going details, then I will tell you there that how this one work. Okay. So see, this one is the thing. So lysosome term is clear, suicidal bag of the cells. Now the point here to know about what? Vacuole. What I'm saying? Vacuole. See, I'm not going in details, but here just I'm going to write about the vacuole. Vacuole is other cell organelles, and you all know that the vacuole is smallest in animal cell. See what I'm saying? It is smallest in which one cell? It is smallest in animal cell. But large in what? large in what? Plant cell. But large in what? Plant cell. Clear? So see with the help of diagram. I'm going to make a diagram so that's you make clear here that this one is what? Plant cell. See what I this one in the plant cell, the outer membrane is what? Cell wall. Getting? And inside that is this one is what? Cell membrane. This one is what? Cell membrane. Getting? And this cell membrane having what? Wait. This one is what? Vacuole. This one is what? Vacuole. Correct? And the periphery region, what I'm saying? Periphery region. Periphery region contain what? Nucleus, my dear. See. This one is what? Nucleus. Nucleus is which one type? Yes, you say this way. Okay, double layer membrane. Here I'm going to make a layer membrane. There is no need to be draw here double layer. Okay, but you know the nucleus is what? Double layer membrane. See, this one is what? This one is what? Nucleus, my dear. Clear? So now I'm going to label here. I'm going to label here. See, this one is the outer wall known as what? Cell wall. Do you know that the cell wall is made up of cellulose? Correct? See, this one is what? In yellow color. This one is what? Cell membrane. Or you say plasma membrane. Right? This one is what? Vacuole. This one is what? Vacuole. And this one is what? Nucleus. Getting my point? And here what? Dense fluid. The name of the dense fluid is known as what? Cytoplasm. This one is what? This one is what? Cytoplasm, my dear. This one is what? Cytoplasm. Getting my point? So see, what the difference between here, the vacuoles in animal cell. See, check here. This one is what? Animal cell, and here is what? Plant cell. Here the diagram of what? This one diagram of what? Plant cell, my dear. Okay. So what the difference here when I'm saying about the vacuole? See, here the vacuole, the size of the vacuole is what? Smaller. Here the size of the vacuole is what? Small. So see, this one is the small size of vacuole here, but while here in a plant cell, the size of the vacuole is what? Bigger. Okay. This one is the first difference. See, inside the vacuoles, there is also fluid. That very fluid is known as, you know what the name of the fluid? The name of the fluid is known as what? Cell sap, my dear. What I'm saying to you? You know? Yes. The fluid, the name of the fluid is what? Cell sap. Wait a moment. Here, the name of the fluid is what, my dear? Cell sap, I am saying. Getting my point? So see, if I'm saying you about the vacuole is present in both plant cell and animal cell. This one is right. But here the little bit difference is here that in a plant cell, the size of the vacuole is bigger in comparison to the animal cell, right? Mostly the space occupied by the vacuoles in a plant cell, while in animal cells, the size of the vacuole is smaller. So that's why they doesn't perform any specific of their functions. Getting yes or no? Getting yes or no? So say this one point. Say this one point. Here the vacuoles, and there are different types of vacuoles, my dear. Number one is what? Food vacuole. Second one is what? Contractile vacuole, my dear. So see. See what I'm saying? Here I'm going to tell you about the vacuole. That is, see, I'm going to write here what? Vacuole, which is responsible to what, my dear? capture the food material. Whenever the food goes inside the cell membrane, then it forms what? A food vacuoles. Just to take an example of amoeba. Getting? So see, this one is the diagram of what? Amoeba, right? And this one is the what? Nucleus. So whenever the food touches the wall of membrane, this one, that is what? Cell membrane, my dear, correct? And this food, whenever goes inside the cell, then it form what, my dear? Food vacuole. See, this one is what? Food vacuole. This one is what? Food vacuole. So what I'm saying here, the cell membrane formed as a what? Food vacuoles, and whatever the digestion, the all the digestion will take place inside the food vacuoles, my dear. Getting? So see, so there are two types of the food vacuole. One is what? Contractile vacuoles, gas vacuoles, sap vacuoles. There are different types of vacuoles, my dear. Okay, so here is which one type of vacuole? Here is what? Food vacuoles. Getting my point? Yes. Now the time to know here some little bit about the what I'm saying to NCRT. So let's check NCRT also, my dear. So see this one point. Wait for a moment. Wait for a moment, my dear. Here I'm what? Making a PDF of cell chapter. Okay, that is given in NCRT. Clear, dear? See this one. Here, what are the things? Unit 3, cell structure and function, my dear. Okay, this one is the chapter. Am I right? Sir, clear? See, hope so this thing is visible to all of you. So this one is visible. Yes. Tell me yes or no. Yes, sir. This one is visible. No? I'm going to make you zoom so that you can see clearly. Okay. Let's see. Yeah. Yes. Hope so this one thing is clear. Now see what I'm saying here. Unit 3 in cell structure and function. This one is what? Unit 3 structure, cell structure and function. So what here we all know that. But see in this, so biology is what? Biology is what? Study of living organisms. You know all that. The biology derived from what? Greek words bios and logy. It means what? Study. So study of life is known as what? Biology, my dear. So that's why I'm saying here the biology is the study of living organisms. So here the detailed description, their form and appearance only brought out their diversity. You all know that the diversity, you know the meaning of diversity, the variety of life present on earth. So all that life, whatever we know, that all they are made up of cell. So it is a cell theory that emphasizes the unity underlying this diversity of forms idea. You know, that is the cellular organization of all life forms. Cellular organization. So all the life will organize from which one? Cellular. Getting? So that's here. Uh, in whenever we go through in a details, then we know about this all things, how this cellular organization will form, because you all know that cell group together to form tissues, tissue group together to form organ, and this organ group together to form organ system, and this organ system group together to form organism, my dear, right? Now the point is here that whenever I'm telling you about the cell theory, so cell theory is created a sense of mystery around living phenomena. You know why I'm saying that cell theory created a mystery around living phenomena? That is physiological and behavioral process. So this mystery was a requirement of integrity of cellular organization. You know, cellular organization for living phenomena, my dear. Right? Now, the point is here. See, this one is the scientist, no, what? G.N. Ramchandran. This one is what? G.N. Ramchandran, my dear. And this G.N. Ramchandran, that is what? Outstanding figure in the field of protein structure, double helical structure, or the founder of the Madras School of Conformational Analysis of biopolymer. He discovered the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed to conformation of protein through the use of Ramchandran plot rank among the most outstanding contribution in structural biology. Yeah. Getting? So this one is the very important thing to know here, that the G.N. Ramchandran, an outstanding figure in the field of what? Protein structure, because we all know that we all are made up of cell, and cell is what? Made up of cell membrane. The outer covering is what? Cell membrane. In an animal cell, that very cell membrane is composed of what? Protein, my dear. So if I'm saying here that they're composed of protein, you know what? If I'm saying they're composed of protein, and this protein structure was the founder of whom? G.N. Ramchandran, my dear. So see, Ramchandran graduated in the year 1942, my dear, at the top ranking students in BSc honors physics course of the University of Madras. He received a PhD from Cambridge University in 1949. So while at Cambridge, Ramchandran met Linus Pauling and was deeply influenced by his publication on models of the alpha helix and beta sheet structure. Getting? That directed his attention to solving the structure of what? Collagen fibers. Getting? So this one is autobiography about the G.N. Ramchandran. So there is no need to be know here. But the thing is here to know what? See, this one chapter, that is what? Chapter 8, cell, the unit of life. So here the thing is to be clear. What is cell? What is cell is here clear? Yes, sir. So cell is what? Structural and functional unit of life. Unicellular organisms are capable of what? Independent existence. And this independent existence and performing the essential functional of life, my dear. Clear? So anything less than a complete structure of a cell does not ensure independent living. Yes, this one is very important line. So anything less than a complete structure of a cell, it means what? It does not ensure the independent living. So whenever I'm saying the independent living, when they have what? Capable of independent existence and performing the essential function of life. So when any life perform any essential function, when? Then when they have a capability to exist independently. Getting my point? Exist independently. So hence, cell is the fundamental structural and functional unit of living organisms. Getting my point or not? See, what I'm saying here, I'm going to repeat here also. When I'm going to say that the cell unicellular organisms are capable. So unicellular organisms are capable if independent existence, and what? Second, performing the essential function of life. Clear? So anything less than a complete structure of a cell does not ensure independent living. If the cell is what? Not a complete structure, or complete structure, not well defined structure, then how we say that the cell exists independently? Yes or no? The cell exists independently whenever they are capable what? To perform a basic function, essential function of the life. So that's why I'm saying here the cell is the fundamental structural and functional unit of all living organisms. Clear? Now the scientist is here. The name of the scientist, Anton van Leeuwenhoek. Yes. And this Anton van Leeuwenhoek, my dear, the scientist who gave about the living cell, my dear, who discovered what? Living cell. But what about Sir Robert Hooke? Before you have to tell that Robert Hooke discovered cell. So which one cell Robert Hooke discovered? Robert Hooke discovered dead cell, my dear. That is in 1665. But this Anton van Leeuwenhoek discovered which one? Live cell or living cells, you say. So see, here the Anton van Leeuwenhoek, the first saw the describe a live cell, my dear. Getting? So this Robert Brown, after that the Robert Brown, Robert Brown later discovered what? Later discovered what, my dear? Nucleus. Getting my point? Nucleus. So nucleus is what? Nucleus is a brain of cell. Where it is located? It is located in at the center in animal cell, while periphery at where? In a plant cell, my dear. Getting my point? Yes or no? So this invention of the microscope, its improvement leading to the electron microscope revealed all the structural details of the cell. So before the invention of microscope, we doesn't have any idea about any cell organelles, my dear. Okay. So as the scientist Anton van Leeuwenhoek invented the microscope, so its improvement leading to what? Knowing about the structural details of the cell. Getting my point? Now, second thing, that is what? The cell theory. Cell theory. Yes, I was told you here's a little bit moment earlier that about the cell theory, that is in mind map. So cell theory is what? In 1838, see, in 1838, the scientist Matthias Chaldean, and Matthias Chaldean is what? German botanist, my dear, examined a large number of plants and observed that all plants are composed of different kinds of cells. Getting my point? See, again I'm saying in 1838, the scientist Matthias Chaldean, that is a German botanist, examined a large number of which one type of cell? Plant cell. What I'm saying? What I'm saying, dear? Plant cell. And this plant cell are composed of different kinds of cells. And these different kinds of cells unite together to form what, my dear? Tissues. Getting? At about the same time, the another scientist, my dear, the name of the scientist is what? Theodor Sion. See here, this one, Theodor Sion, in 1839. Clear? And this Theodor Sion is what? A British geologist. Matthias Chaldean is what? What? Botanist. And here is what? Geologist. So this Matthias Chaldean and Theodor Sion studied, basically Sion here, studied different types of animal cells. And that different type of animal cells reported that the cells had a thin outer layer. And you know the thin outer layer today known as what? Plasma membrane. What I'm saying? Plasma membrane or cell membrane, or you say the selective permeable membrane. Getting my point? Yes. So this selective permeable membrane only allow the useful substance to be in and out. So that's why it is known as selective. So here, Matthias Chaldean and Theodor Sion proposed the hypothesis. On the basis of Schumann hypothesis is that the bodies of animals and the plants are composed of what? Cell and product of cell. See what I'm saying? I'm saying here, my dears, at whatever the things given by the scientist about the cell, that is Matthias Chaldean and Theodor Sion, one the botanist, another one is geologist. After observation of that two scientists, on the basis of, you say, the Sion proposed that the cell, either the plant cell or animal cell, they are composed of product of what cell? So Sion and Chaldean and Sion, you say, together formulated the cell theory. Together formulated the cell theory. But this theory, however, did not explain as to how new cells were formed. Getting my point? Here I am saying that Matthias Chaldean and Theodor Sion both formulated the cell theory. This one is correct. But this theory, however, did not explain that how the new cells were formed, my dear. Getting? So then after, in 1855, the name of the scientist is Rudolf Virchow, first explained that the cell divide a new cells and form from pre-existing cells, that is omnis is cellula. Getting? This was the Latin word. He modified the hypothesis of what Sion and a Skaldian to give a cell theory final set. That's why cell theory is understood today in the form of two. That is, number one is what? All living organisms is what? Composed of cell and product of cell. And second one is what? All cell arises from pre-existing cell. So see, this one thing here I tell you in a mind map. See, here this one thing I am telling you in what? Mind map, my dear, about the cell theory. Correct? Correct? Here I am giving one example of that is what? A zygote cell. This one is a new cell. And this very new cell arises from pre-existing cell. The name of the pre-existing cell is what? A sperm and ovum, my dear. Getting? So this cell theory given by the two cell theory. Number one, Matthews Skelton and Theodosian, that all cell arises from all living organisms is composed of cells. And Rudolf Virchow, all cell arises from pre-existing cells. So this together is known as what? Known as what? Cell theory. Getting my point? Getting my point? Yes. Now the time is here to know. After that, the lysosomes, vacuole, endomembrane system. Okay. Now, the thing is here to be know here about that endoplasmic reticulum, ER of two types. What I'm saying? ER of how many types? Two types. See what I'm going to write here. I'm going to write here what? ER of what? Two types. One is what? SER, and second one is what? RER. Clear? So SER means what? A smooth endoplasmic reticulum. See what I'm saying? A smooth endoplasmic reticulum, my dear. And RER, rough endoplasmic what? Reticulum. Getting? So ER how many types? Two. One is what? SER. Second one is what? RER. So when I'm saying you about the SER, that is what? A smooth endoplasmic reticulum. And when I'm going to tell you about RER, that is what? Rough endoplasmic reticulum. But now the question is here, when we say the RER, we say RER whenever it contain ribosomes. It contain, what I'm saying? It contain ribosomes. Where? Ribosomes where? On the surface of what, my dear? ER. See, this one is suppose ER. It is a tubular structure, and here the what? What is the bigger unit? Second one is the smaller unit. This one is here. So this one is present. This one is what? Ribosomes. These all are what? Ribosomes. Getting my point? So this one is the ribosome, which is present where? On the surface of what? ER, my dear. This one is what? ER. Clear? So when the ribosomes contained on the surface of ER, then it is known as what? RER. This one is what? RER. RER. But whenever the ribosomes is not present on the surface of ER, then it is known as what, my dear? SER. Getting my point? So see, this one is what? Make here. Here, this one is also tubular like a structure, and this tubular like a structure is only what? SER. Why I'm saying this one is SER? Because they do not contain ribosomes on their surface, right? See this one. So if I'm saying here about the RER and SER, this one is the main differences. So cristae, vesicles, tubules, they are different types of ER. Okay, when we go in details, then I will tell you there. Let's see here first. Then afterward, the mitochondria. This one is what? Mitochondria. And you all know that this mitochondria is what? Powerhouse of cell. What I'm saying to you? Powerhouse. You know the mean why I'm saying the powerhouse of the cell? Because whatever the energy is stored, that energy be stored where? In a mitochondria. Suppose here, whenever we take any food, after the breakdown of that very complex food, they store in their body, na? So where it is stored? It is stored where? In a mitochondria. So that's why I'm saying here that the mitochondria is what? Powerhouse of the cell. Getting my point? See, mitochondria has double layered membrane. One is inner membrane, second one is what? Outer membrane. Clear? So inner membrane having a crystal poses F0 to F1 particles medium. While in outer membrane, that is the what? Outer membrane. See, plastid. Second one is what? That is what? Plastid. This one is what? Plastid. Other cell. And this plastid is only present where? In a plant cell. This one is only present where? In a plant cell, my dear. Getting? And this plastid is how many types? Three types. One is chloroplast, second one is chromoplast, and third one is what? Leucoplast. Getting my point? So if anybody can say you about the plastid, then what you say? Listen here. The plastid. Clear? Number one. Chloroplast. See what I'm writing here? Chloroplast. See, chloro means what? Green. Chloro means what? Green. So here the green pigment present in a plant is known as what? Chloroplast. Plast means what? Pigment. So see, if I'm saying here that the green pigment, what I'm saying here, green pigment contained in a green plants. So then the very pigment is known as what? Chloroplast. Second one is what, my dear? Chromoplast. What I'm saying you? Chromoplast. Chromo means what? Colored. Chromo means what? Colored. See, and plast means what, my dear? Pigment. So the colored pigment contained in our plants is known as what? Chromoplast, my dear. Getting? And third one is what, my dear? That is leucoplast. What I'm saying? Leuco. Leuco means what? Colorless. Colorless. And plast means what, my dear? Pigment. Plast means what? Pigment. So the colorless pigment, my dear, there is not any color there. Getting my point. So there are three types of plastid. One is what? Chloroplast, chromoplast, and leucoplast. Is that clear? So leucoplast means what? Take one example. I'm just giving an example of the root. I'm giving you example of potato, the outer scary parts. Is there any color containing there? No. So that's why there is what? Leucoplast. The color of the root? No, that is colorless. There doesn't any contain pigments. So that's why it is known as what? Leucoplast. So whenever we go through in the details, then I will tell you about these all things in very details, my dear. Getting? See. So here the three plastid is clear. One is chloroplast, chromoplast, and leucoplast. Right? Now, third one is what, my dear? That is what? Ribosomes. And you all know that ribosome is what? Just I tell you. Ribosomes is known as protein factory of the cell. See what I'm saying you here. See, this one is the ribosomes, na, which is present on the surface of ER, or some other ribosomes which is also floating in cytoplasm also. But this ribosome is the main responsible for what? Protein factory of the cell. What I'm saying here? Protein factory. Why I'm saying here the protein factory? Why I'm saying here protein factory? You know why? Because this ribosomes mainly is responsible for the formation of protein, my dear. Synthesization of protein, my dear. So that's why I'm saying that the ribosomes is known as what? Protein factory of the cell. What I'm saying? Protein factory of the cell, my dear. Getting my point? Yes. This ribosomes is what? Pre-planta americana. You know that is in pre-plantana americana. That is what? Prokaryotic. Prokaryotic containing how much ribosomes? 70s ribosomes. What I'm saying? 70s ribosomes. While in eukaryotic ribosomes, that is what? 80s ribosomes. So this one is the thing. So prokaryotic, how much are S? 70S. And in eukaryotic, what? 80S. Clear? So what the meaning of S? S stand for Sevdberg constant. Getting? Sevdberg constant, my dear. Clear? But the Sevdberg constant, here the two unit. One is the bigger unit. Second one is what? A smaller unit. Correct? So one is the bigger. Second one is what? A smaller unit. See, this one is the diagram. Wait a moment. See, this one is the diagram. This one is what? A smaller unit, and this one is what? Bigger unit. Yes or no? So here, if I'm saying about in prokaryotic cell 70S, right? So here, when I'm saying about the 70S, here in this 70S, they are combined of what? Two. One is what? Yes, that is 60, and what? 30S. Getting what I'm saying? Yes, that's why this is called 70S. So see, this one is the thing. Wait for a moment. I'm going to tell you through but NCRT also. See, after this one thing, let's see here, an overview of cell. An overview of cell is what? See, whenever I'm talking about the overview of the cell, there's something little bit point here to know that the nucleus, eukaryotic cells have other membrane bonded distinct structure. This very distinct structure called what? Organelles. Here what I'm going to tell. Organelles na. So see, endomembranes, endoplasmic reticulum, ER, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. These all are what? These all are the distinct membrane bonded organelles, my dear. So here, this one is prokaryotic cells, lack such as membrane bounded organelles. Yes, there's some also organelle that which doesn't containing any membrane. Yes, non-membrane bonded organelles, found in all cells, both in eukaryotic cell as well as prokaryotic cell. So here the point is very important to know, my dear, that the cell either having the word membrane bounded or some organelles which do not contain any membrane bounded organelles. You know, within the cell, the ribosomes are found not only in the cytoplasm. Ribosomes is not found only in the cytoplasm. Yes, ribosomes also found in within the two organelles also. That is in what? Chloroplast in plant cells and mitochondria in mitochondria and on rough ER. See, rough ER, rough endoplasmic reticulums, ribosomes contain, and other cell organelle also, that is chloroplast in plant cell and mitochondria also, my dear. So animal cell contain another non-membrane bounded organelles called centrosome, which help in cell division. See here, this one is a very important point to note here, that when I'm saying that the some cell, not in some cell, in animal cell, they also contain membrane bonded organelles, and some are not membrane bounded organelles. So which one is not membrane bounded organelles, my dear? Centrosome. And the centrosome is only present where? In an animal cell. What the function of the centrosome? The main function of the centrosome is what? To help in cell division. Any division of the cell. Getting my point? So this one is the important thing. Now, here, the something that is given about the shape and size of cell. See, cell have different, see what I'm saying, cell differ greatly in size, shapes and activities. Just I'm going to take some example of this one thing, but not I'm going in details. For example, suppose take mycoplasma. This one is the smallest cell. Yes, sir. And what the size of this smallest cell? That is 0.3 micron meter in length. While bacteria, my dear, see here the bacteria could be how much? 3 to 5 micron meter. Getting my point? See this one, the diagram. See, this one is the diagram. This one is diagram of RBC, red blood cells. What the shape of this RBC, my dear? Round and biconcave. This one is white blood cell, WBC. What the shape? This one is amoeboid. This one is what? Columnar epithelial cells. So what does say? Long and narrow. See, this one is the different structure. Here the nerve cell. What does? Size branched and long. This one is what? Trachea, which is found in plants. Elongated cell. This one is what? Mesophyll cells. And mesophyll cells is this type? Round and oval, my dear. Round and oval. Getting my point? So see. So the largest isolated single cell, that is an egg of ostrich. What I'm saying? Egg of an ostrich. And this one is egg of an ostrich is what? Largest isolated single cell, my dear. Getting my point? So among whatever the multicellular organisms, it's like RBCs, about how much? 7 micron meter in diameter. Nerve cells, longest cells are also vary greatly in their shape also, my dear. Not only the size, but also vary in their shape also. What they vary? They may be disc-like, just example is RBC. Second one is what? Polygonal, columnar, cuboid, thread-like structures, and even irregular structure, my dear. Irregular structure, which one? So take an example of WBC, amoeba, amoeboid shape. Getting my point or not? So the shape of the cell may vary within the function they perform, my dear. Okay. So whatever the function they perform, they become vary. Right? This one chapter is very interesting, my dear. Yes. So when we go through in details, then here I'm not going to read only this NCRT like. I will show you there that how the thing is to be there. But here I'm minded to be say here only about some thing that is not goes in detail. Why? Because this one is the mind map. And this mind map only tell about the little bit information that whatever the thing is written here. Getting my point or not? So see, after that, here the things is written what? Uh, cytoskeleton. See, cytoskeleton is here. Other cell orders, cilia and flagella, which is basically found where in a prokaryotic cells. That is in bacteria, flagella, paramecium, cilia. Also, centrosome and centrioles. These all are centrosome is what? Centrosome is what? Uh, Not membrane bonded organelles, which is mainly responsible for cell division. Centriole help in the what? Formation of what? Spindle fiber during cell division. Now, the nucleus. Nucleus is very important, my dear. The nucleus is known as what? Nucleus is known as what, my dear? Brain of cell. So when I'm saying this one is brain of the cell, and why I'm saying the brain of cell? Because this one is the nucleus is responsible to perform what? All the basic metabolic functions, my dear. Whatever the cell perform any functions or metabolic functions, they are regulated by whom? That is nucleus, my dear. So that's why I'm saying to you, the nucleus is what? Brain of cells. Yes or no? So here, this one is very important to know, my dear, that about the nucleus. So I'm going to tell you about this one nucleus. See, here I'm writing a little bit information. Okay, little bit information, not in details, but this one point is very important to know, my dear. So this nucleus, it is discovered by, first what I'm writing here, it is discovered by whom? The scientist. The name of the scientist is what? Robert Brown, my dear. But I'm written here, Robert Brown. So this Robert Brown discovered what, my dear? Nucleus. So see here that in nucleus, this one is what? Nucleus. And this nucleus is what, my dear? Double layered membrane. Here. This one is what? Double layered membrane. This one is the nucleus. In animal cell, it is located in centrally, while in plant cell, it is located where? In the periphery. Wait for a moment. Getting? See. Inside the nucleus, there is another spherical body known as what? Nucleolus, right? And inside this nucleolus, there is a what? Dense fluid. The name of the dense fluid is known as what? Nucleoplasm. See, this one is what? This one is what, my dear? Nucleoplasm. See what I'm saying? This one is what? Nucleoplasm. See, this one is nucleoplasm. Here, one thing I'm going to make, that is a thread-like structure. This thread-like structure is known as what? Chromatin fiber, or you say the date one. Now I'm going to level here. I'm going to level here this one thing. See, here this one is what? Outer membrane. This one is what, my dear? Inner membrane. And this outer and inner membrane known as what? Nuclear envelope. What I'm saying here? Nuclear envelope. Getting? And this one is what? Pore. This one is the channel. This one is the what? Channel, or you say the pore. Okay, this one is the minute openings. And this very pore is known as what, my dear? Nuclear pore. What I'm saying? Nuclear pore, my dear. Correct? And this one is fine thread-like structure. This fine thread-like structure is known as chromatin fiber. What I'm saying? Chromatin fiber. And this one is what? Nucleolus. If one, nucleolus. If more than one, that is known as nucleoli. Nucleoli. Why I'm saying the nucleoli? Because nucleoli is plural. If one, then it is known as what? Nucleolus. So this chromatin fiber, my dear, and this chromatin fiber is a fine thread-like structure which is floating where? In a nucleoplasm. And this chromatin fiber become condensed to form chromosome. You know, here the term, what I'm writing here, chromosome. Chromosome term is very important to know, my dear. So when I go through in a details lecture, then I will tell you about that, all the things. Okay, but here I'm going to just through in a brief, my dear. So if anybody can ask you about the chromosome, then what you say? It is condensed part of chromatin fiber. See what I'm writing here. It is condensed part of chromatin fiber. Chromatin fiber which formed during cell division. Which formed during when, my dear? During cell division. Getting? So chromosome is what? Condensed part of chromatin fiber. So this one is the chromosome. This one is what, my dear? Chromosomes. And this chromosome is the condensed part. And this one is what? Centromere. This one is what? Centromere, my dear. And this one is what? Chromatid. This one is chromatid. This one is also what? Chromatid. And this two chromatid attach at a point. That very point is known as what? Centromere. Clear? So see, these all are the overall things about the chromosomes, and further the chromosomes be divided into four parts on the basis of the attachment of the centromere, that is metacentric, subcentric, ecocentric, telocentric. Yes or no? See, this one is mind map. Is given here or not? Yes sir, this one is given. But here given, dear, see here. One is what? Metacentric, when the centromere is at the middle of the two chromatid. One is what? Sub-metacentric. One of the chromatid is bigger than the other. Sub-metacentric. Third one is acrocentric. One of the chromatid is much bigger than the other. That is called acrocentric. And fourth one is what? Telocentric. It means that centromere is at the apex of the chromatid. So whenever I'm going in details of this very video, my dear, this one chapter is very, very interesting and important chapter. That is cytology. Yes or no? So this one is only the mind map. So here I'm just only give you a small little bit information about this very topic. Okay. So whenever go in details, then I will tell you about these all things, my dear, in a very, very detailed manner, my dear. Okay. So thank you for watching. Thank you. Have a nice day. Take care.